degrees Celsius
Wow.
up to 23 degrees Celsius. So I just said from 8 to 23 Celsius, they'll develop normally, because that's kind of what they were saying. That
Yeah, I guess so. Okay, it's been a while.
yeah, so I got that straightened out, and I have my presentation ready for them, but I have to do a real sales job.
Bradley, I tried the update command on your file, and
Okay.
unfortunately, it updated some of it.
What do you mean? Only some of them, or?
Only some of them, yes.
Oh.
Okay, so I have to do a double check.
Okay. Yeah.
Okay.
But I found a couple mistakes and I fixed them. So the comments are.
Okay, yeah, that's why I'm working from yours. But if there's anything I can't figure out, I'll let you know.
All right, that sounds good. Yeah.
Okay. That I'm actually finished the book last night.
Oh, okay, good.
I've got to fix this chapter and one other chapter, and then I can send it off.
Oh, good.
Okay.
Yeah, that sounds great.
It's only been 3 or 4 years.
Yeah.
The thing that took the most time is compiling pictures of all the archaea I could find, so that I could try to be objective about which ones were polygons.
Oh, yeah.
And I finally got the figure, so that those for which I could get figures, 41% had some polygonal cells.
Okay. Yeah.
Which is incredible.
Yeah.
Okay, so anyway, that's kind of the major conclusion of the book, and the source of the major speculation is that maybe life started that way.
Oh, okay. Yeah. 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 That's great.
So anyway, it was rather satisfying.
Right. All right. Well, that sounds good. That looks like a pretty good book.
It's going to be a bit controversial.
Yeah.
Like the chapter you wrote with me and 2 other chapters both suggest that origin of life could occur at equilibrium.
Yeah.
Which is completely against what everybody else says. You got to have an energy source.
Yeah.
So that's one of the strange conclusions.
Yeah.
But the problem is these people don't have statistical mechanics training. So I think we've shown that fluctuations from the average can, if you've got a big enough ensemble, be quite large.
Right. Right.
You remember that was one of the points of the ergodic theory.
Oh, right.
In a room, it's possible all the molecules will rush to one side of the room.
Yeah.
All the air molecules. It's a very low probability, but it's there.
Right.
So if I start gasping for breath, I'll worry about it.
Hello, Susan. How are you doing? I heard you were still going through your presentation for your program.
Yeah, I'm trying to find a new candidacy project, and I put something together for imaging axolotl eggs with optical coherence tomography, because it works quite well. They've tried it, and at least you can get the upper part of the egg and image the blastocele. So it is imaging blastoceles. It's like its volume, its shape, at what temperature, or what time, and at what density. And it would make a paper.
Yeah.
I showed it to Dick, and that was his comment to choose a publisher of your choice and get the thing published.
Yeah.
So I'll see if it goes over with the professor. He's not a fan of axolotl eggs, and it's partly because he doesn't know about them very much, and I would have some trouble.
Do you have a real biologist? Susan, do you have a real biologist on your committee?
Dr. Zhang and Dr. Franca. Dr. Franca's with dentistry.
Dentistry,
okay. And so he would understand what this was about.
Yeah.
The other fellow's an electrical engineer, so I have to keep it. I have to explain stuff. It's a good thing I have to explain stuff because actually Dr. Sharif isn't a biologist either. So I'm dealing with two electrical engineers, one biosystems and one dentist.
Okay, good luck.
So the dentist, I'm pretty sure he understands the biology, but and also the biosystems problem.
Yep. All right. Okay.
Yeah. Yeah, it's not really the axolotl egg-friendly territory there. I mean, you know.
It's an axolotl egg. It's slimy, okay?
Yeah.
You can put it under your microscope.
Slime is not an engineering term.
Right. Yeah, what did I say? The axolotl egg will naturally adhere to the surface of the slide if its gel is left on it.
Oh.
Yep.
Well, you've got gel in there, but not slime. That's not.
You have to make an analogy with crazy glue.
Analogy to what?
Crazy glue.
Oh, crazy glue. Yeah, it really adheres. Anyway, my suggestion, put it under the microscope, just out in the air, take images, about 5 of them, slices, put it back in the water, and then do the next one.
Okay, as long as it doesn't dry out.
Yeah.
Yeah, no, it won't. It takes about a minute to get one slice. So 5 minutes out in the air is not going to bother it if it's in its gel.
You could also control the humidity if you have to.
Yeah, but I'll let the experts on the optical coherence tomography baby their equipment because they have to take it at an extreme angle to get down to the 1.5 millimeters. So I'll let them deal with that. I hope to be in there helping take images. They're going to have 3 shifts as far as I can tell, because 8 hours is about as much as I can do. So 8 hours, somebody else takes over, and then I'll do 8 hours.
Yeah, do you remember our funny episode where we slept overnight in the lab and took pictures, and at the end of the weekend, I think it was the weekend, we figured out that the eggs had all died.
Oh, yes. Yeah, I'm hoping for a viable batch. I sent for a new female from, she's in Ontario from Hamilton.
Okay.
And she's shipping her tomorrow, and she'll arrive on Thursday. We hope if she makes it.
Okay.
She called her a Lucy, a leucistic, white with dark eyes. But anyways.
White with dark eyes.
Yeah. Anyway, I hope we're getting this new female and that she's healthy. And then I won't be mating brother and sister together, which is begging Saskatchewan to sell me a pair, and they said they don't usually sell pairs. I'm going, well, I have a pair, and they're a brother and sister, and I want to break this up. It's like I paired a brother and sister together for the eggs that I took images of that I gave you because I didn't have anybody else, and they mostly died. They were not really viable. So, yeah. So I don't want to do that.
Okay.
But they still went through their cell divisions, but they didn't live beyond neural tube closure, if that.
Okay.
But I got some nice images.
See, Bradley, we always had trouble mating the males.
Oh.
So what we would do is we would take them for a car ride. They would barf, and then they'd mate.
My technique is plunge them both after they've been isolated for a long time. Plunge them both into really cold water.
Oh,
with
yeah,
plants.
that probably works too.
And that often works too.
A ride on a country road might work.
Yeah, and if the plunging in cold water doesn't work, put another ice cube in the water and take for a bumpy ride. I don't know. Anyway, I'm into this again. I hope that he accepts my proposal because it would be fast. I get eggs. I spend 48 hours, and it's a fast experiment. So, anyway, that's what I'm up to right now.
Okay. Well, that sounds busy. Yeah. Yeah. Thanks. Yeah. Sushmanth, hello.
Hi, Bradley.
How are you? All right.
I'm working on making the graphs actually, which you have mentioned.
Okay.
Comparison between DeepLab V3 model, SAM model, UNet model, and our cell SAM model. I have fine-tuned SAM and give it as a cell SAM. Micro SAM was taken by some other people, so I kept as cell SAM name, and I'm creating the comparison between all these models actually. I'm working on this thing. I'm trying to train the data of our C. elegans data and create graphs about that. Right now I'm working on that. Bradley, could you just create 2 repos in DevoLearn, empty repos? I could start pushing the code to the main repo because my deadline is approaching on October 24th. By that time I will push all code to the DevoLearn organization, what I worked on full GSOC today. Right now it is in my repos actually. So if you open in 2 new repos, I will just contribute to them directly from here.
All right. Why do we need 2?
2 repos. One for fine-tuning SAM on microscopy images. Another one is DevoNet.
All right.
One repo name would be DevoNet. Another repo name would be CellSAM. If these 2 repos are ready, I will just push the code. And coming to Hacktoberfest, Bradley, I have created the readme.md file, but I need to host it somewhere. Where should I host it? The thing.
We'll talk about that in a minute. I've got some things ready for that. So, yeah, I'm glad to hear that you have it though. So,
okay. I have created a basic GitHub README file. So if someone comes there, just read the README and they will understand what is the task. My idea is each people need to take some around 10 images minimum, up to 20 images maximum, and they need to create the annotations for that data set. And we will mention that this would be next GSOC project for them for creating cell lineage analysis by these fine-tuned models. And do you have anything to ask about Hacktoberfest or the project I have mentioned?
I don't know. I was thinking about it a little bit, but I haven't been able to. Well, I think we have some of the details you just mentioned what they were. So, I watched the video and I saw that, and then I created some I created a repo, and I'll show that in a minute. And then I guess we need to store the data. I guess you're going to make data, and we need to store it somewhere.
I have the data set. I'll just push it directly to the repo. I have the data set right now itself.
All right.
After this meeting, I'll push the data over there. So each people takes a certain number of images and they will annotate it. There are around 5,000 images actually.
Okay.
How many as they can do it. They can push it. That's the only thing.
All right.
Show me the repo. I haven't seen the repo actually. That's the problem.
Yeah, we'll get to that in a minute. So, how do we know if someone takes some data? How do we know what they've taken? Are they
I have taken a CSV file. Let me share my screen and show it to you.
All right.
Share screen. Is this? Could you?
Yeah, see it.
Okay. see, Bradley, these images, one was segmentation maps, one was features images, and these are named actually to the numbering. If you see the numbering of these images, 0, this is a TIFF file, right? It has so many slices.
Yeah.
Around for each TIFF file, I got around 34 slices, I think so. For this 30 slices, it has came. For F0, 30. So there is a numbering actually, F0, 18, up till 30 it is there. And if you can see this, there are some empty images in this thing. So I have created a CSV file where images are not empty. They have some cells over there. So they can directly mention like from this file to this file I have taken this, I'm working on this thing. They can create an issue in the repo, so can and they will work on that issue. Just a minute. See, Bradley, could you see this CSV file? I can extract this actually.
Okay, see it, yeah.
This thing, this CSV file, these are the images where cells are there actually. If you see F010.png, from here only the images started actually. Sorry.
Okay, yeah.
From here only the images started. Before that, there are no images in this thing. So they can see this CSV file and they can directly like write from this number of image to up till this thing. I am making the data set and they can create a issue on the GitHub. So no one will work on those number of images, and other people can take from below. This goes in a sequentially. Just we have printed only 15. There are 5,000 images actually.
Yeah.
So yeah, that would be useful. I will create the CSV file and images also, and they can mention an issue in a certain format that these number from 010.png to 020.png, some guy is working. After that, 0_21.png to from next set of photos, one guy is working. Say they can create issue and keep a pull request. When they have completed their work, we can close the issue and we can close the pull request also.
Okay.
I will take care of that thing, hosting the data and how the process should go. But before that, I need 3 repos actually. One for my GSOC, 2 for my GSOC projects, 1 for the Hacktoberfest support.
Yeah, yeah. Okay, I mean, the only thing I have a question about with the CSV file is, like, if people look at it, they can see what's available. Well, they can see all of them, but then they just take like a subset, and then people
from CSV. Okay, from CSV file they can decide. See, Bradley, in this thing,
yeah.
There are some empty images, right? In this, no cells are there. We don't need to annotate it.
Okay.
So I have created where the cells are present, in which images the cells are present. So from this image, actually, you can see there are some cells over here, and here, and next one also has images 1, 2, 3. So from the CSV file, they could mention create an issue in GitHub, like showing, see, I'm working on these number of images, these set of images. So please, other members don't work. We'll create some structure. According to that, the issue will be assigned to them, and they can work on that.
All right. Yeah, I'm just a little worried that it's like people are going to get confused and work on the same images or not exactly know from the issue. But on the other hand, well, I'll show you what I was thinking, but I don't think that would be much better. So, I mean, you know,
otherwise we will create the issues actually, Bradley.
Okay.
Mentioning these number of images are there, they can work. For each person, we will give 10 images, so they can work on that issue. They can close the pull request in the parallelly. You understand it, right?
Yeah.
We'll create set of images. On how many people are coming, they could just ping me as in the Slack channel. So we can create an issue for them and assign it to them. They will work on this for whole month. That would be fine.
Okay. Yeah.
I will take care of the creating issues over GitHub and assigning to the people. So that won't be a problem.
Okay.
If one issue is assigned to one people, they will complete that, and we will not give to other members those images. That would be, I think, works right.
Okay. And then what would happen after they work on them? Like, what is the output?
Output is data annotated file and a CSV file will be generated. So according to that, they will push those CSV files and the images into the repo.
All right, and that would have to be into a folder, like that would have to be into a directory, or they would just create their own directory.
We will keep in a separate folder which are annotated.
Okay.
Yeah, I will try to create. Please give me a collaborator access to these three repos. One was Hacktoberfest, CellSam, and DevoNet.
Okay.
I will create a specific template for Hacktoberfest, and I will create some rules over there. And according to that only, whoever wants to contribute, they can work on that issue.
Okay, that's good. That's good. Thank you.
Sorry, I was a bit 2 days late. I was working on my models actually, GSOC project, so I couldn't concentrate on it.
That's okay. Yeah. So thank you for the update.
Is it possible, like, can we discuss it tomorrow further along? I will work on this template and accept, and by tomorrow it will be ready. So whoever you start keeping this thing, advertising it, Hacktoberfest, we are participating in it. We can discuss it tomorrow if you want any further changes over it, what I have implemented.
Okay. Yeah. We could do it over Slack, but just yeah.
Okay, that would be great.
Yeah. Okay. Well, thank you, Sushmanth, for the update. So yeah, Hacktoberfest starts. Well, I guess it started with the month of October, and it continues through the end of the month. And so it's formally something that's sponsored by GitHub and a couple other organizations, and they put this together to encourage people to contribute to organizations. We've participated in the last few years in different ways. We don't usually give people like swag if they do a certain number of pull requests. Usually, participating orgs, if someone issues 5 or more pull requests, they get like a T-shirt, and they can actually do this. We have to put a label on the repo, and they can like use that as proof of work to GitHub to get the T-shirt. We don't usually give people anything. Sometimes we link it to next year's GSOC, and that makes it attractive for people to contribute. So we'll set it up, and then we'll make it go live formally as a Hacktoberfest repo, and people can work on it. And they can work on it after October 31st. It's just that's usually the official period is the month of October. So that's great. We'll see what we can get in terms of people working on it, and we'll have to work out the system of distributing data because I don't know what'll be best for people, but we'll figure it out. Hello, Jesse. How are you? Looks like Jesse's here. All right. So let me go over what I had in terms of setting up our Hacktoberfest. So I've opened, and I'm going to probably rename this repo, but I want to give it a name that's not just Hacktoberfest. I want to give it a little bit better name. So when I came up with this model annotation, it's probably not the best name, but I'll need to give Sushmanth editor or full access to this. I just kind of worked it out last night. Basically, I have a little bit of a template here for setting up the README file. So Sushmanth, if you want to put your README that you have in here, you can just put it in as like the information, the instructions. I just put some basic instructions. The idea here would be, you'd go to this repository, you go and I guess we can distribute, have, media is just for the images in here, but data sets is the one that maybe we can use. I don't have anything in there right now, but we can use that. Oh, no, I have things in here. So data sets are where I know that Sushmanth asked me about data that has like the labels for cells and C. elegans and positions, which are in 3 dimensions, X, Y, Z. And we have them in these files, these CSV files. These are from our DevoWorm repository in the DevoWorm GitHub. And so I just brought them over here and put them in data sets. So if you go in here, you can see that they have the developmental cell ID. This data set has developmental cell ID, the different XYZ coordinates. These are normalized. And then I need to probably put together a file that describes what these are. And then this is the size of the cell. Actually, it's from a normalized median. So I have to describe what these are. Then we have, let's see. Okay. Then we also have data on the sort of adult cells, mapping that to developmental cell ID. So the adult cells are the ones we're used to thinking about when we think about, the nomenclature for the adult. We have the adult cells. We also have some of the lineage identifiers. And then this is the developmental cell name here. And then this is the description. So we have a description for each cell too. So if we annotate things, we have functional descriptions. We have a developmental cell name, which is like the cell ID when it's in the embryo. And then a adult name, which is the cell that it's going to become when it's an adult. And because our C. elegans has this deterministic mode of development where all of the developmental cells will have a defined fate right from the time that they divide. So, or for the most part, anyways. And then the final one is, we have some more data here with developmental cell. This is the absolute position. Actually, this is, I think, also normalized. And then this is the volume. This is the division time in minutes. So this is where, you can go through an image or a movie, and if there's a timestamp, you can match it up to a certain time in the sequence. So that we have all those data. We'll have to work on it a little bit. Maybe work with people who want to contribute. But that's what we have. And so that's quite a bit of annotation data, I think. So yeah, we have the MIT license in here and then the README. So I'm going to invite Sushmanth to this. We need to figure out kind of how we want to do this. I guess we can set up issues where we have, maybe we can set up an issue template as well. I'll set up an issue template where I don't know how we want to set up the template. Maybe we can put in some questions like, what are you working? What files are you working on? Things like that. So I don't know what kinds of, I don't know, maybe if you have ideas, Sushmanth, on what kinds of issue template we want to set up, what the questions would be or the statements.
Actually, I was thinking, Bradley, I will create a template and mention in that template, like, if someone is working on certain set of images, other people can't work, and we will assign the issue, the person whoever wants to contribute to it. And I will create, I don't have anything right now in my mind, but I will definitely create a template by today night and I will upload it here. And another thing, I have a small doubt regarding the cell naming actually.
Okay.
How we know the cell, it is a, A cell, or there are like certain number of these things, right? A cell or E cell, T cell. How could people know whether it is like, how could they know the ABALA and this cell belongs to this name exactly? I think we need to create a tutorial for them, read the documentation, then they should know, they should annotate the data.
All right. Yeah,
I mean, AB is there, Bradley. For example, AB is there. How could they know, like, at what position it is the cell naming it as AB?
yeah. So, I mean, there are a couple ways that can be done. So, one is to observe the cells kind of dividing and, like, trace the lineage from, like, a two-cell state. But if you're dividing up the data, that doesn't make much sense necessarily. Then we have these positions here, which are sort of centered on like a median of 0. So they actually, if you have an image, they should be in a certain part of the image. So they should be kind of like, if you kind of go from the center of the image, which is 0, you can go down to those coordinates, X, Y, Z, and then you can find maybe the general location.
I'll share my I'll share my screen.
Okay.
Basically, how we name this thing. See if this cell is there. How we know which part of cell is named as A cell, B cell, etc.
Is this C elegans?
An example.
Is this C elegans or not? This is not C elegans.
Yeah. They are C elegans.
Oh, okay.
They are C elegans 3D data set, but there are only 4 cells.
Huh.
Yeah.
Like that. Yeah, I see. What's the orientation of those images?
Orientation, I didn't get you, Bradley. Could you?
So, like, if we look at the left-hand side of the image, what is that? Is that the anterior end or the posterior end?
This is the posterior. This is the anterior.
Okay. So yeah, we probably want to make like tell people how this. So that would be like I guess it'd have to be flipped sort of. I guess to do the annotation, you'd need to flip it 90 degrees to the right, so the anterior is on the top. And then and then those coordinates should be like if you go from the center of this image and then go like XY, you can just take the XY coordinates and kind of find the direction. So if it's like X positive, it's at the top, in the top half. If it's posterior, it's at the bottom half. And then for Y, it would be like, I think the positive would be right and the negative would be left. So I'll have to write that up.
Is there any materials I could see about this naming kind of thing? I will attach to the row.
Well, this is just kind of like we're making this up from the data. There is like the interactive lineage tree and things like that, but it's the
naming wouldn't be that much important. At least the bounding boxes would be useful, I think so.
Yeah.
Bounding box and actually most of the data sets we have right now, those don't have a bounding box or whatever data set they use. So at least bounding box is useful to fine tune these models by giving prompt. Right now, when I was working on the SAM model, the most problem I have encountered is I have the data set, I have the segmentation maps of it, but I don't have the bounding box. Actually, to fine tune these on these large language models, we need something prompt like bounding box, a point, or a text prompt at least. So this at least they could annotate with the bounding box. That would be useful for us.
All right.
So I will think through that and I will create a template by tomorrow. That would be ready by tomorrow. Just give me some time. I will go through these things and this. Okay, let's see. I will create the template. Just give me a collaborator access to the Hacktoberfest.
All right, I will. Yeah. And then I'll create those other 2 repositories as well, so we have those.
Okay.
Yeah. All right, so that's what we're getting ready for that, so that should go live, fairly soon, and we can start getting people involved.
Okay.
All right. Yeah. Great. So that's great. So we'll be working on that over the next several weeks. Hopefully that leads to like a GSOC project next year. And then, of course, our GSOC projects are due on the 23rd. So keep working on those. All right. So actually, I'm going to talk about this paper here. This is a C. elegans paper that just came out recently. This was September 14th. This is a nice paper on C. elegans connectome and looking at different behaviors. The title is Brain-Wide Representations of Behavior Spanning Multiple Timescales and States in C. elegans. So the idea behind this paper is that you have these different circuits in C. elegans that are involved in basic movements. So for example, there's a forward movement circuit and then there's a backwards movement circuit. And people know those pretty well, and it's like maybe about 8 or 9 neurons a piece. And those are pretty easy to like trace out and figure out which neuron is active for, which kind of thing that you're looking at. So it's almost like a car transmission or a set of bike gears, where you set it in one state and then certain neurons respond and it produces that behavioral output. This is a little bit different. This is actually taking into account larger scale networks. And so this is not just like a small circuit, it's bigger networks that help to modulate a lot of the activity in C. elegans movement. So, you have these basic movements forwards and backwards, omega turns and things like that. But you also have it happens at different speeds. Sometimes the worm is going over different types of media, like a gel or like soil, and it's needing to control its movements in different ways. And so this requires a larger set of neurons. So this is a group of authors here did the work. The graphical abstract basically shows that they're recording behaviors and neural activity brain-wide, and then they're adding in quantitative behaviors. They have this probabilistic encoder that predicts neural activity, so the behaviors are put into what they call an encoder model, which is a machine learning model that analyzes a lot of the data that's coming out of this analysis. So brain-wide neural data plus quantitative behaviors is a lot of data that you don't know what direction it points. So you want to use a model to sort of get some potential answers out of it. So you put the behaviors under this encoder model. You fit it to a posterior, which means you have this, this sort of Bayesian model of prediction. And then you have this measured and predicted neural activity. You have two traces of time series. The measured is the blue, which is what we've measured from the worms, and then the predicted is the orange, which is what was predicted from the encoder model with the fitted posterior. And those kind of, you can see they overlap in this example. And then the next step then is to look at like what kinds of behavioral encoding information can be mapped onto the connectome. So in C. elegans, we know what the connectome looks like, we know what the cells are, we know what the connections are. So we can take the neurons in their different classes. So, we have the neuron identities, we know what the functional aspect, what they do, and then we can sort of look at the encoding features. And then we can put this onto a connectome diagram and show that, like, for these different colors, those are different states at different times. So basically each cell in these networks do something different at different times. So depending on the movement that's needed or the movement that's happening, we have these different, the neurons do different things in this network. And so you can actually generate connectome diagrams for this part of the connectome that alternate based on the different behaviors. So like speeding up and slowing down or going over a gel or whatever, the state of the neurons will be slightly different for each of those cases. So you see the neurons and the synapses are connected in this way. And then finally, we have defined neurons show encoding changes upon state change. So the state change of the sort of the behavior corresponds to the encoding changes of the neuron. So we have, like, for example, heat stimulation induced behavioral state change. If you expose a C. elegans to heat or like an elevated temperature, its movement behavior will change. And when its movement behavior changes because of the heat, those circuits that are sort of controlling forwards and backwards still are they still operate, but the modulation of those movements is different. And so you can see that these dynamic this connectome diagram with the different colors, it's a little bit different in terms of how they encode this. So the highlights here are that they took simultaneous recordings of brain activity and behaviors in C. elegans. They built this probabilistic encoder model that describes how each neuron encodes behavior. They mapped the results to a connectome, and they didn't map it to the entire connectome. They picked subsets of the connectome. And these are above and beyond the little circuits that we use for basic movement motifs. This yields an encoding atlas of C. elegans' nervous system. So we're not just looking at the neurons and the connections, are looking at how they encode things. So one neuron can have multiple encodings, and then those encodings connect together to generate this kind of what they might call a representation. Although that word tends to be loaded, and we don't want to use that freely too much, but that's basically what they're doing. And then the defined neurons that are in this network flexibly change encoding in a state-dependent manner. So as the behavioral state changes, these encodings change, and it's flexible depending on the context of what the worm is doing. So this was published in Cell. These are the authors. So the summary reads, changes in an animal's behavior and internal state. So this is where you have like a movement behavior, and then its internal state is the thing that's shaping that behavior. So if a worm is in trying to navigate around a space where there's scarce resources, or whether it's trying to migrate from one end of a gradient to another, a chemical gradient, or it's migrating across the gel, those are all things that have a different internal state attached to them. So changes in these behaviors and states are accompanied by widespread changes in activity across its brain. However, how neurons across the brain encode behavior and how this is impacted by state is poorly understood. So we don't know exactly how, even in the C. elegans brain, a lot of this behavior is encoded and how it's impacted by state changes. So, we kind of make up states of neurons. We make up states of sort of global states of behavior by measuring it and observing it, saying that's a turn, that's a slowing down or speeding up. But sometimes, the states are pretty distinctive. So, all these things are encoded in the brain, in the connectome, but we don't really understand how those changes translate. We recorded brain-wide activity and the diverse motor programs of freely moving C. elegans, and built probabilistic models that explain how each neuron encodes quantitative behavioral features. By determining the identities of the recorded neurons, we created an atlas of how the defined neuron classes in the C. elegans connectome encode behavior. So they built this atlas. Many neuron classes have conjunctive representations of multiple behaviors, meaning that they kind of are using the same neurons. Those representations are sort of hinge upon one another. So, like, for example, you have a network of maybe 20 neurons, and it can produce a number of sort of representational states. If it's speeding down or slowing up, slowing down or speeding up, or it's like, I don't know, maybe making a complex three-dimensional list to the worm, moving to its right side or to its left side. Those are things that are all in those connectome parts of the connectome, and they all sort of you can reach one from the other. So that's what they mean by conjunctive. So the same neurons can represent multiple behaviors. You can go from representation, the representation can change based on the behavioral state or substate that you want. And I say you want because it's like the C. elegans. So it's a little hard to, the C. elegans isn't thinking about doing it as far as we know. It's just that the neurons actually represent, things that are possible. Moreover, although many neurons encode current motor actions, so a lot of neurons encode motor actions. A lot of neurons that we have in the annotations, for example, are very broad functions. Sometimes they're just kind of like motor neurons, or they have some general function, but they actually have a lot of like sub-functions, and they can be refined in different contexts. So, when we say it's a motor neuron, but it does it does a lot of things that are kind of hard to figure out what exactly they are, because you can't observe the worm in every possible set of actions. So, although many neurons encode current motor actions, others integrate recent actions. So you actually have these sets of neurons that do sort of different things in time. So some of them are encoding current motor actions. So like if the worm is moving forward, those neurons are encoding forward movement and the sort of the variations in forward movement. Some of them, however, are integrating recent actions. So they're looking at the past of the organism, and they kind of sample that space, and then it gives a representation that is useful for maybe a future action. And so, in a lot of neural representations, you need to have both a current state and maybe a past state to produce a future state or a prediction. So, not every action is equally probable, given like the current state and then the set of past states. And that's in line with what we think about behavior. If you've experienced a certain set of things, you're more likely to go in a certain direction than another direction. And that, in fact, that's why they have representations, is because they need to have that information so that they don't, do something wildly inappropriate for the environment. So if you're in a gel, for example, you want to be able to, the C. elegans wants to be able to navigate the gel without too much trouble, and to do it smoothly and things like that. Changes in behavioral state. So if you have a worm that's changing its behavioral state quite radically, are accompanied by widespread changes in how neurons encode behavior. And we have identified these flexible nodes in the connectome. So they're actually nodes that are flexible with respect to these encodings. Some are more flexible than others, and they identify those in the paper. This is kind of reminiscent of the Rich Club neurons paper, where they identify several neurons in the C. elegans connectome that are highly connected, as opposed to others that are less well connected, and those have functional consequences. And they organize sort of the control systems of the connectome. So we have, Rich Club neurons. We also have these flexible club neurons, I guess you could say. Our results provide a global map of how the cell types across an animal's brain encodes its behavior. And we start with a very small connectome of 302 neurons. And then maybe this is informative for larger connectomes. So we have two comments.
What is the title of the paper?
Okay, so the title is...
Can you just show it again?
Brain-wide representations of behavior spanning multiple timescales and states in C. elegans. And that's in Cell Journal. It was from this year. 2023.
Okay, thanks.
Yeah, no problem. It looks like a bunch of people from MIT and Leeds in the UK. So it's, okay. So if you go through the paper here, so they talk about the nervous system. They talk about some of these behaviors. C. elegans that generates a well-defined repertoire of motor programs, locomotion, feeding, head oscillations, defecation, egg laying, and postural changes. And those are all very stereotyped, so they're represented in the connectome pretty faithfully. They're not just spontaneous movements. C. elegans expresses different behaviors as they switch behavioral states. So animals enter sleep-like states during development after intense stress. Awake animals exhibit different foraging states like roaming versus dwelling. And adverse stimuli induces sustained states of increased arousal. So they have all these different states, or all these different, not just movement states, but things related to movement states. So you get this need for like representations. In C. elegans, it may be feasible to decipher how behavior is encoded across an entire nervous system and how this can flexibly change across behavioral states. And so previous studies have identified that some C. elegans neurons reliably encode specific behaviors. So we look at neurons AVA, AIB, and RIM. Those encode backward motion. And these are these circuits that I talked about where they're very small circuits that really you can look at like different neurons, and they always produce the same behavioral output. It's sort of like almost like an electrical circuit. It's that reliable. Another one is AVB, RIB, and AIY and RID. So there are four of them. RIB and RID versus AIY and AVB. Those encode forward motion. So you have backward motion and forward motion. SMD encodes head curvature, and HSN encodes egg laying. So you have these different circuits with very small numbers of neurons. They always encode these stereotypical behaviors. You get, in some of the neurons, you get corollary discharge signals, which propagate information about motor state. So already you're starting to see in these very small networks or these very small circuits that there's like auxiliary information being sent out to the rest of the nervous system. And so we get these different things that need to happen in movement. We need to understand, or the worm needs to get information about proprioceptive feedback. So if the worm moves, it needs to know if its movement is successful in the environment, and then it needs to keep integrating that. So there are other neurons that are involved there. So what they're trying to do is they're trying to figure out like if these small circuits exist and there are larger cells that are supporting these small circuits, then what are all these neurons doing in terms of representing the actual set of information that it needs to make these movements? And so they actually were able to do this with some really fancy neuroimaging for C. elegans. So I think they're looking at some sort of calcium imaging, not entirely sure. And then, they're able to characterize what's going on in the different neurons. Then they can use a machine learning model to extract information. So they wrote software to automatically extract calcium traces from videos. They use a 3D UNet first to locate and segment all neurons at all time points. They register images from different time points to one another, and use clustering to link neurons' identities over time. And these are called, so it says STAR methods. So they're using the STAR method, I guess. To test whether this accurately tracks neurons, they recorded a control strain expressing NLS-GFP. So they're actually looking at tracking neurons with GFP signals. They have these, so they can actually find the cells that are involved in these things. So that's one way to do that. Make mistakes in linking neurons' identities would be obvious here, since the green fluorescent protein or the GFP signal would fluctuate in a neural trace if time points were sampled from different neurons. So they're actually able to use the level of GFP and look at the neural trace and kind of match those up to find the cells. So we just talked about like identifying different cells. This is how you do this functionally. But of course, in the nomenclature, it's a little bit different. But in any case, that's what they had to do to get the networks together for this. And then they basically looked at behavior, and they looked at some of these stereotypical behaviors and how some of these networks are contributing to regulating that. So let's see, this figure is, I'm trying to find the legend here for this. Legend on the next page. Okay, it's down in the bottom in the footnotes. So fraction of neurons per animal that encode the indicated behaviors. This is A. B has fraction of neurons per animal that encode 0, 1, 2, or 3 of the behaviors. Let's see, so let's look at the figure. So I was interested in this E, actually, but so A and B and C and D are here. They're kind of looking at the percentage of neurons at B and the number of behaviors encoded. So you can see that most of the neurons in these sampled parts of the connectome encode either 0 or 1 behavior. That's 40% each. So that's 80% of the neurons. And then 2 behaviors encoded are about 20% of the neurons. And then 3 or more, I guess 3 behaviors or maybe more are encoded by maybe a very small fraction of the neurons. And so you see this matches kind of how the percentage of neurons here, velocity is encoded by about 50% of the neurons. Head curve is encoded by about 20%, and feeding is encoded by about 15%. So you get this breakdown in terms of different behaviors, how many neurons are involved, and then different types of behaviors encoded by single neurons. You get the same distribution, where very few neurons are encoding a lot of behaviors. They're encoding specialized behaviors and so forth. So this E is what I want to get to here. If we go back down to the legend, distributions of how neurons encode the indicated behaviors. Neurons are categorized based on their tuning curves to each behavior. And again, you see star methods for more. Example tuning curves are shown above. The prototypical tuning curves for each category are shown. So rather intriguing here. We have velocity is up here, and then the neuron types are down here. So we have, for example, forward and reverse. We have forward rectified A, which is 23%, forward rectified B, which is 20%, and analog forward, which is 9%, slow, which is 3%, and fast, which is 1%. So they're able to break down the neurons in this network in terms of these different aspects of the behavior: forward, rectified A, rectified B, analog forward, slow, and fast. And so you get these curves, which are the activity of the neurons. So some of them are quiescent, and then they start to ramp up. Some ramp up and then are quiescent. Some are stable, and so forth. And then if you go for reverse, you have reverse rectified A, reverse rectified B, and analog reverse. So you get these different components of different behaviors. Then for head curvature, you get a similar thing where you get forward rectified ventral, and then for dorsal, you have forward rectified dorsal. So these are different parts of head curvature, ventral or dorsal, and then so it's moving up or down basically. The head in C. elegans, they move their head up and down, for reasons that involve sensory acuity and if they're exploring their environment. So you get this sort of different type of control for your head curvature than for your velocity. And then you get feeding neuron types. So you get feeding related behaviors when feeding is activated versus when it's inhibited. So you see these different components here. So that's nice that they can map all that out. And then they don't really show, they show a lot of, I guess this is like a UMAP of embedding of all neurons of 14 animals, where proximity indicates encoding similarity. Here we projected all points from each neuron's C, P, N, E, M posterior. So this is from those posterior distributions. This is kind of hard to read, but basically, you'll have the neurons that are closer together do the same thing. So it's kind of, this doesn't tell you very much. This, I guess, is what we're looking at. We're looking at these different components of the behavior. So like the feeding activated neurons have an affinity. The feeding inhibited neurons have an affinity. Long and short time scales have an affinity with each other, or with long and short neurons respectively. So long time scale with long time scale, short time scale with short time scale. You have dorsal and ventral behaviors. So just like we saw in that figure, and forward and reverse. So basically they're able to sort of, from all these different measurements, a large number of measurements, are boiling this down to 2 dimensions. And this is the UMAP method. So it's like a dimensionality reduction into this bivariate space. And then they're saying, what are the similarities between the points based on all that data being boiled down to 2 dimensions. And so this is, it's different than like a PCA, but it's basically a dimensionality reduction. And then you look at the commonalities of the things projected into the space. So that's then they get to the networks where they actually have the cells that are identified, and they have these muscles that serve as sort of the place where the outputs go. So you have these neurons here in head curvature during forward. So you can see that there's this color bar which tells you kind of the state of the neuron, what it's doing, and then the connectivity, and then it's projecting to these muscles, which are then controlling the movement. So you can see that as these states change, the muscles are modulated in the outputs, and then the behavior is modulated as a result. And so you actually have this very large network here where you have head curvature during reverse, and that's linked to a subnetwork of variable coupling, and then that's linked to some other intermediate neurons. And then you have the pharyngeal connectome, and then some cells in the locomotory part of the connectome. So these are connected into larger networks, and you're able to look at the modulation of each subcomponent, but you can also look at the modulation in the global component here. So this is, so there's probably much more here to talk about, but I'm gonna, I think we got the point of the paper. Basically goes through, and they do a lot of good work on kind of grounding, a lot of the behaviors that are generated by the connectome into these kind of, different types of changes. So you have state changes, and you have, intermediate modulatory states and things like that. And they're able to actually pull this apart using both calcium imaging and behavioral assays. So the discussion reads, animals must adapt to their behavior to a constantly changing environment. How neurons represent these behaviors and how these representations flexibly change in the context of the whole nervous system was unknown. They employed these methods, these distinct tunings and timescales and levels of flexibility that map onto defined neuron classes in the C. elegans connectome. So that's that paper. So Sushmanth had to leave. Thank you for attending, Sushmanth, and I'll attend here to attend to the Hacktoberfest stuff soon. So are there any questions or comments?
Interesting morphology where you get all those neurons together. It's sort of almost looks more random than the epithelial tissue that I've been looking at. It kind of looks like I don't know, more of a bird's nest
Yeah.
than something that's got a stripe running through it that organizes things.
Yeah.
There's an order to it, but it's
Yeah, it's not like linear. Yeah. I guess that's a good question. Like, why is the connectome not linear? Like, what is the advantage of having things kind of jumbled together like that? I mean, if you thought about
maybe
like,
it's just the way it grew.
oh, maybe, yeah. Well, I mean, there's this idea that like the connectome has to be sort of optimal in terms of its wiring, which is a hypothesis, but the idea is that, you want to have efficient routes from neuron to neuron in a circuit or in a network. And, if you look at a connectome, it always looks like it's the standard, what they call the hairball network, which is like it's all jumbled together and you have like a lot of loops and things like that. But if you think about how like neural systems are regulated, they need a lot of feedback and they need a lot of this sort of flexible connectivity. So you have, things coming in from different places. You need to maybe have different modes of output. And so that's why you have that. It isn't a linear response like a muscle. It's actually quite different than that. So
yeah, and it's then there's glial cells that are filling in between too and keeping the neurons alive. So
yeah. Yep. Okay. Any other comments or questions before we go?
Just wondering how on earth I would model that.
Well, people, model, like they do biophysical modeling for neurons, but like the question is like what kind of modeling would you use? Would you use biophysical modeling, or what? Like maybe if you want to model the growth of the tissue, that's a different set of models. People have it. There's been some multiphysics modeling on brains. I know, like for different, it's kind of a new area where they use multiphysics models to model different aspects of like connectomes. So they'll look at like the cellular physics. They'll look at like the signaling activity, the biophysical activity, other things like that. And they'll put it all into one model. And maybe it predicts things differently than if you just use the biophysical model.
Okay.
Yeah.
I'll stick with epithelial tissue.
Yeah.
For now, it's like more straightforward in a way.
Yeah.
Although it's got a thin film on the bottom of it, so that's always interesting too.
Well, thanks for attending, and see you next week. I'm going to talk about a few more papers. These papers are organized loosely around the theme of physiomes and the molecular brain. So the first paper is on this idea of the cell physiome. So we've talked about, earlier in the meeting, we talked about C. elegans connectomes and how those are sort of have variable representations of behavioral states. And then the question came up from Susan, and Susan and myself had a conversation about it. Why do brains look so messy if you look at the connectome? And so, like with muscle, you have a linear power output, a linear force output, and the muscle is striated, or other types of muscle that have this linearity to them. So that's different from the brain, where you have a lot of loops and tangled connections and things like that. And some of that is regulatory, but some of that is, we're not really sure why. So this paper talks about the cell physiome. So if we want to predict single cell biology, or if we want to have a framework for representing single cell biology in a computational framework, and this is at the level of physiology, this is not at the level of individual cells that are sort of doing things for themselves or on their own, we need to rethink how we kind of model some of these things. So in that conversation, we talked about multiphysics models, where you have different aspects of the physics that are being modeled simultaneously, and it gives you a much different answer than if you model different processes in isolation, such as biophysical models, force models, and so forth. So this paper talks about the cell physiome. What do we need in a computational physiology framework for predicting single-cell biology? So this looks like a group of Australians who did this paper. So the abstract reads, modern biology and biomedicine are undergoing a big data explosion, needing advanced computational algorithms to extract mechanistic insights on the physiological state of living cells. So we have big data sets. We're generating data, physiological data, gene expression data, as we saw in the last paper, voltage data. So we have all this electrophysiological data associated with that. So there's a lot of information there, and they're huge data sets, but they're not very well integrated into insight. And so, we can do things like take all the data and put it into a UMAP and show these pretty pictures, but they don't really tell us much about the mechanisms. And that's really kind of where we want to go, because we want to understand, say, for example, how a physiological system works, how it can be controlled by, different mechanisms within the body of the organism, how the different systems interact, these sorts of things. We can't know that unless we have like another layer on top of the big data. And so this is what they're arguing for. We present the motivation for the cell physiome, a framework and approach for creating, sharing, and using biophysics-based computational models of single-cell physiology. So they're modeling at the level of the single cell. If you think about their modeling framework, their compartment is a single cell, and they're building these compartments, and they're putting together a systems approach. So this is something that, so if we go to the board, we can see that like what we basically have in modeling are these systems which are integrated. So if we have the organism, which we use as this box, we're going to be very nondescript about this. We have systems. So we have different systems here. And these systems are, they're kind of defined by function. So we have like the nervous system, the endocrine system, and so forth. And so they all have a function. They all have a boundary set up around them that's defined in that function. So for example, if you have a bunch of cells that process information, we call it a nervous system. It's connected to a brain, a central nervous system, or a peripheral nervous system. We call that system. If it's connected also to the endocrine system, then the boundary is a little bit fuzzier, but we still maybe consider it to be part of the nervous system or part of the endocrine system, depending on how it's connected. And people have done these studies without big data. They've done them with small data, with experiments, and they've kind of delineated where these cells belong. So one of the aspects of the physiome or big data is that it operates on the assumptions of small data, but it advertises itself as being this new set of insights that can only be gotten through big data. So it's kind of hard to kind of get to those new insights without kind of being beholden by these kinds of system boundaries and things like that. So a systems biology approach could redefine these boundaries, but that's actually not where most studies operate in terms of the container of the basic unit of analysis. So in this case, what they're arguing for are individual cells. And these are our containers, our basic units of analysis. So everything that we might model would be run inside this container. this container would contain all of the models and programs, and then we would just replicate those containers across, say, like a system. So the central nerve, so in the last paper we had a connectome. That connectome were neurons. They were individual containers of neurons. Each container had one neuron. And then we had a bunch of containers in a network that were connected in different ways. You can have in the C. elegans connectome, you can either have like gap junction connections, electrical connections, or chemical connections, which are synaptic connections. And so these are all sort of defined by, again, our experimental data that we've done. Someone had to create a connectome. The draft connectome was gotten through actually looking at microscopy and analyzing that microscopy. So that's not necessarily high throughput data. These are just kind of, data that there's a lot of hard grunt work that gets involved in making these data sets. So this is what we have for a connectome. So these are our containers. They're networked into this formation. They exist within these larger systems partitions, and then the organism is a collection of systems that interact themselves in different ways. So each of these containers on the bottom right or on the bottom left are neurons. Those containers are replicated across the nervous system. That nervous system has maybe some configuration of containers in a network. And then that system, or that sort of subsystem of the organism is maybe connected with other subsystems in different ways. And so that's where we have our full organism model. So it operates on that principle. And so there are a lot of questions as to how do we make these containers maybe more effective? So if we model a neuron and we say that's sort of the level of analysis, is that really the best level of analysis? Or maybe there's something going on sort of at the circuit level. Or maybe there are a lot of interactions between the nervous system and other parts of the organism that are interesting. But anyways, this is the way it's organized traditionally. This is how they're doing it here. So they're building these biophysics-based computational models. So they're taking each container, the traits of the sort of the characterization of this container, of the cell, is largely biophysical. So we don't care about gene expression so much. We don't care about the behavior of the cell, how it migrates. We just care about the biophysics. And so when we take a cell in biology, we're typically abstracting away, we're operating at a certain level of computational extraction. So we're taking away a lot of things that maybe are irrelevant to the question we're asking, and we're building a model based on those attributes. So a biophysical model, for example, would involve ion channels, maybe the production of action potentials, maybe the different types of genes that are expressed relevant to the biophysics, but not necessarily a gene regulatory network that controls, say, the state of the cell, or whether the cell is a stem cell or some sort of precursor cell or a mature cell. A couple weeks ago, I talked about how to model an organoid, and a brain organoid. And one of the problems you have is that the brain organoid isn't always in the mature cell state. It's not in that terminally differentiated state. It's often in these stem cell states or transitional states where you have neural precursor cells, or something that maybe isn't quite like our traditional knowledge of what a neuron should look like. So sometimes you have neurons that maybe behave like a motor neuron, but they don't have the full complement, the full biophysical complement of a motor neuron. So this is something that is an assumption that we make in modeling, and sometimes for whatever we're doing, can be right or wrong. So using examples in calcium signaling, bioenergetics, and endosomal trafficking, we highlight the need for spatially detailed, biophysically based computational models to uncover new mechanisms underlying cell biology. So this system is very sort of squared off, but you can see that there's a spatial relationship here. If I were to build a model more along the lines of, say, C. elegans, and I gave it a shape that looked like C. elegans, I would have like something like this, where we have a worm body, and we have different systems that are squared off so that those squares are sort of mapped to different components of the system. So if we have the square is like the CNS of the worm, this is the egg-laying region of the worm, I guess, the germline and that sort of thing. We have and we have a smaller box that would contain sort of the other parts of the connectome. And then we have maybe like the head, the sensory organs, and so forth. And so we just partition up this form into different boxes, and it's spatially distinct because we can tell the CNS from the reproductive tract. And so we have that spatial relationship. We preserve that spatial relationship. We preserve the partitioning of these cells, and then we can actually model it from there. It's very important information in the embryo, especially because the embryo, these things aren't spatially distinct yet. Sometimes they're very close to one another, whereas in the adult worm, they get pulled up into different parts of the organism. So they can build spatially detailed biophysically based computational models to uncover new mechanisms underlying cell biology. We review progress and challenges to date towards creating cell physiome models. We then introduce bond graphs, which is a specific type of representation, as an efficient way to create cell physiome models that integrate chemical, mechanical, electromagnetic, and thermal processes while maintaining mass and energy. So this is what we were talking about earlier as well. Is that they don't call it a multiphysics model. They call it a model that is holistic, that integrates chemical, mechanical, electromagnetic, and thermal processes. So it's not truly a multiphysics model, but it's getting there. It's incorporating different processes simultaneously so that you can look at these different aspects of how a compartment is affected by like changes in temperature or changes in chemistry or some mechanical stimulus or whatever. This is all the case while maintaining mass and energy balance. Bond graphs enhance modularization and reusability of computational models of cells at scale. So they're putting this first thing here into a bond graph, and we'll see what that looks like. This is just a set of boxes that I drew. We conclude with a look forward into steps that will help fully realize this exciting new field of mechanistic biomedical data science. So they talk about the Physiome project, which is an international effort to develop mathematical and computational and modeling frameworks to represent, understand, and predict human physiology. So they want to build biophysics-based multiphysics, coupling fluid mechanics and solid mechanics to describe coupling of blood flow and cardiac mechanics. That's one of their sort of principles. So they do actually mention multiphysics here explicitly. What you're doing basically is taking the solid mechanics of tissues and the fluid mechanics of tissues. So this is something Susan's interested in with respect to some aspects of cell migration and other types of aspects of mechanics of the phenotype. And so this, if you think about the fluid mechanics and solid mechanics of the connectome, for example, those look very different than a model of individual cells that are sort of in a network that's very abstract from any of the biology. So this is a novel, sort of way of doing this for, say, like the connectome. But it's absolutely necessary to get a lot of the information we need for modeling the organism. And then multi-scale modeling of physiological processes. So that means modeling at different scales. That means taking these containers that are the cells and then plugging them into a larger framework of systems. The second is models that are anatomically detailed to account for key structure-function relationships. So we're doing this, giving anatomical detail to spatial detail that I mentioned here. So we have our different systems in the worm, and they're spatially distinct, and we can actually look at their spatial relationships to see what the structure and the function might be. So for example, if we know that there's a nervous system and there are sensors and effectors, which are muscles and sensory organs, we can put those together and say this is where the worm is kind of moving towards. The worm is moving with its muscles. The muscles are in a certain place to maximize its ability to move through different media. And so we can answer those kind of questions. Models defined using model sharing standards, which is, of course, just standardization for good practice in science. And then model predictions that can be validated with experimental data. Although in many cases, modeling drives new measurements. So we can actually take our models, validate it with experimental data, like we saw in the Connectome paper with between observed behaviors and predicted behaviors, and then we can actually develop new measurements because we're actually sort of asking new kinds of questions by building a multiphysics model. The Physiome project has led to the development of detailed computational models of many organ systems, including cardiovascular, musculoskeletal, respiratory, and digestive systems, as well as the reproductive system. So they're able to pull these together with clinical imaging and omics data, which is the measurement of gene expression and protein expression and things like that, to characterize the patient-specific organ-level physiological state. So they're interested in medical applications or interested in the organ-level state. So these containers are combined into an organ, and they're predicted from that. Okay, so let's take a look at what bond graphs are. So they talk about bond graphs in the abstract, and they don't really mention. I mean, maybe a lot of people aren't familiar with what they are. But this is from Figure 3 in the paper. So in panel A, this is a bond, and this represents power transmission with power variables U and V. So these are different variables that they've defined earlier in the paper, and so they're kind of defining these in different ways in this bond graph. They're exploring this relationship. So this is where they have copower variables U and V. So you can see that there's power transmission that is equivalent between the two variables. In B, you have all these arrows pointing to the middle, and 0 U, and then V is equal to 0 with the sum. So they're actually equal, but they're so the B is a 0 node junction with a common potential U, and the corresponding mass or charge balance, sum of V equals 0. So U is the potential and mass or charge balance is V. So these are two different aspects of a system. These are also in balance. In this case, in C, we have an imbalance. We have a one node junction with a common flow V and the corresponding energy balance equation, sum of U equals 0. So it's an imbalance here. So we have this sum of all the charge balances or the masses coming together, and we have a potential that's greater than that. Then in D, we have this more complicated relationship. We have a bond graph representation of a simple reaction. So this is a reaction where you have different components of U and V. This is just demonstrating the balance and sort of how they can co-vary. This is where you have different Q components where it's producing different products, and you have these U and V within. So you have 0 U and 1 V, and there's this reaction going on. So it's modeling this reaction in this way. So the bond graphs are really just kind of boxes and arrows models with different components that have to balance or not balance. And then E is a bond graph representation of the reaction between calcium, Ca, and troponin, C, TNC. So you can see that you have these different parts here, these different products, and these different relationships within the bond graph. And so you can see that you have these sort of end states and things that go around. So this is the way the bond graph works. They can become quite complex, but they model a lot of these relationships and reactions. So one of the things you're doing with bond graphs is you're looking for mass balance and energy balance. So you're actually looking at how these systems are regulated using these different, so these models are from sort of chemical engineering type equations. They're based on the Michaelis-Menten equations. And so this is where this is coming from. So a bond graph is useful for these kinds of specific types of modeling exercises. There are other types of, I don't know how well they work with other types of things that we might be interested in. So as they mentioned, this is something that's useful for kinetics. And so this is, of course, what they're specialized for. Bond graphs have been used to model a wide range of other biological systems. These include metabolic networks, signaling networks, electrophysiological systems, mechanochemical systems. So they're somewhat broad in application and scope, and there's some references here you can look at. Now they've developed an open source Python package called Bond Graph Tools. So you can actually construct bond graph models, construct them and simulate them fairly easily. If you adopt standards within the CellML framework, which is this XML standard for cell modeling, bond graph models can be annotated using biological semantics. So you can add a semantic layer to your models. You can add in annotations. You can merge them into larger models and so forth. One of the things they're trying to get at with this type of modeling is that people in, say, experimental biology often talk about cell processes being expressed and regulated, being involved, being required, or participating in things. And so we take these parts of the process and we assign these sort of terms to them as if we understand what they do. And, like when I say one thing is involved in another, is involved in a process, I can say that if I don't really, I may not understand the mechanism fully. But you can, you use these terms to describe what you think is happening or kind of what it might be happening. It's not a rigorous definition. It's just something that we say because the data suggests it. So my factor being involved in some process. We don't really know how it's involved, but we can say it's involved because it co-varies with the process. And, this is very separate from like statistical or computational definition. So when I say involved or regulated, I'm just saying that it's part of that, maybe part of that system or part of that circuit. And so I don't really have a good rigorous definition of what that means. I'm just kind of making a reach in some ways. Something participates in a process is even more vague because it's there, it's participating, but it doesn't really have, like you can't give it a statistical test or ask if it co-varies or whatever during that process. It just simply participates. Or if something is required, it may be required, but then we need a more rigorous definition of how exactly these things are required. A lot of times in biology, especially in molecular biology, you have things that we think are required for a process, but sometimes it's only in a certain context. So we actually have, sometimes in different model organisms, different results. And so what you can do in this case is build models that are more broad, wide-reaching. You can have more computationally rigorous definitions of some of these things. So the components in a process aren't just involved or participating, but they have a specific connection, they have a specific effect size, and so forth. And we can model these things from data where we can run a simulation to show sort of the full range of how these things happen. The other paper I want to talk about is maybe more philosophical, and that's this idea of, towards the idea of molecular brains. This is in the International Journal of Molecular Sciences. So this paper kind of talks about maybe some of the things that we're doing in single cells in, say, the epithelium, or we have cells in the liver or something like that. They can do things that nervous system cells do. We maybe typically ascribe to neurons, but are also not neurons, and so we want to know why that is. So this is, I guess, a far-reaching idea. We'll see where they go with this. So the abstract reads, how can single cells without nervous systems perform complex behaviors such as habituation, associative learning, and decision-making, which are considered the hallmark of animals with a brain? So neurons are well known for their ability to produce habituation states, associative learning states, and decision-making states. Oftentimes even very simple nervous systems like the C-slugoplegia can exhibit these kinds of behaviors. They're very simple conditioned behaviors. But, aside from like a nervous system, you can see this with other types of cells. So single cell organisms without nervous systems. We've talked about a lot of these single cell models where they not only the single cells exhibit morphogenesis, but they exhibit basic learning and habituation. So, like for example, their paramecium is well known as a model for this, but there are other single cell models as well. Are there molecular systems that underlie cognitive properties equivalent to those of the brain? This review follows the development of the idea of molecular brains, which is just the way they define these brains within a single cell that maybe is driven by some molecular process. It follows the development of the idea of molecular brains from Darwin's root brain hypothesis. Through bacterial chemotaxis, the recent discovery of neuron-like R protein networks in the ribosome. So bacterial chemotaxis are a good example of this type of behavior. Bacteria follow chemotactic gradients, meaning that there's chemical gradients in the environment, and bacterial cells, being single cells, will follow them. And of course, single cells acting collectively in a colony will follow this, and it will affect the behavior of bacterial cells. This can't be in a brain because there are no brains in bacteria. It's just a single cell with molecular interactions going on inside of it. And so hence the idea of a molecular brain. So then they also talk about neuron-like R protein networks in the ribosome. So in this case, you have an organelle where you have some network within it that can actually process information. So this is another example where it's maybe not strictly molecular networks, it's actually some sort of cellular structure within the cell. But of course, it's mediated by molecular aspects. So this is something that it's kind of an emerging idea. I think there are a lot of other papers that talk about basal cognition, which is where you have cognition without necessarily having a fully formed nervous system or even a nervous system at all, just within single cells. Slime molds there's been a lot of studies on that, and other organisms that are maybe exhibiting some of these things. So by combining a structural biology view with a Bayesian brain approach, and this is again using a Bayesian system to sort of model some of these things. So Bayesian system is where you have a prior, you have a process that integrates information, and then you have a posterior. The posterior integrates the currently observed things in the world, where you use a Bayesian model to integrate it with a prior, and then you end up with a posterior distribution which updates the prior. So the cell has to have some sort of memory, at least a limited memory of states. It has to have a way of currently evaluating those states and integrating them. And then it has to have a posterior or an ability to update what it's experienced. So this is why we use a Bayesian brain approach. But we're also drawing from structural biology. So we're drawing from networks within the cell, other types of things going on. This review explores the evolutionary labyrinth of information processing systems across scales. Ribosomal protein networks open a window into what were probably the earliest signaling systems to emerge before the radiation of the three kingdoms. And this is at the very base of the tree of life. So this is something that is a very ancient network in terms of evolution, but also has this very simple structure. Well, ribosomal networks are characterized by long-lasting interactions between their protein nodes. Cell signaling networks are essentially based on transient interactions. As a corollary, while signals propagated in persistent networks may be ephemeral. So this is where you have these signals that are ephemeral. It's not as stable as a nervous system, but it still does the same type of information processing. Networks as interactions are transient, constrained signals diffusing into the cytoplasm. And this is durable in time, so you have things like post-translational modifications of proteins or second messenger synthesis, which are where you have interactions that are transient. They constrain signals diffusing into the cytoplasm, and this is durable in time. So we have these post-translational modifications. We have second messenger synthesis, and these are different, specific examples of things. They're not the same as the nervous system, but they're ways that, this is used in single cells. The duration and nature of the signals, in turn, implies different mechanisms for the integration of multiple signals and decision making. So this integration of multiple signals is very important. You get different chemical signals acting, and they need to be integrated for the cell to make what is called the decision. But what really boils down to is what behavioral state does it need to produce right now? And so, and there may be some factor where it actually can draw from past states. We don't really know. There are aspects of different epigenetic things that are working to sort of guide some of these states. So we actually have maybe memory mechanisms within the cell that are very limited, but they do allow for that sort of memory encoding. Evolution then reinvented networks with persistent interactions with the development of nervous systems and metazoans. So what they're arguing here is that you have networks that are very much not persistent. They're kind of emergent, and they're sort of itinerant. They sometimes they work. They're transient. Sometimes they work in the presence of certain things like post-translational modifications. But in metazoans, you start to get these systems where you get these persistent interactions and persistent memory encodings that maybe last a lifetime instead of maybe 24 to 48 hours. And so you get this sort of, you get the emergence of neurotransmitters instead of peptides, which help this transition along. You think about it as a transition in evolution. You're transitioning from like these ephemeral networks to these persistent networks. The persistent networks being sort of the nervous systems of animals. The transient networks are more, they're more widespread. You have them in animal cells, is what they're proposing, but they're not the main method of encoding information. Ribosomal protein networks in simple nervous systems display architectural and functional analogies. This comparison could suggest scale invariance in information processing. At the molecular level, the significant complexification of eukaryotic ribosomal protein networks is associated with a burst in the acquisition of new conserved aromatic amino acids. Knowing that aromatic residues play a critical role in allostatic receptors and channels, this observation suggests a general role in pi systems in the interaction with charged amino acids and multiple signal integration and information processing. So they talk about how this could be the molecular basis for future computers with organic processors, but there's some really interesting evolutionary lessons here as well. So that's all for today. Thank you for paying attention, and I hope you learned something.